Proteins are the basic functional units of all living organisms, and they're responsible for all biological processes. In our final project, we thought we'd try to apply our newfound knowledge of data science to predict the structures of proteins from their genetically encoded primary sequences. At the basic level, proteins are encoded from simple strings of amino acids. A major question, though, in biology is how to predict three-dimensional structures from these sequences. Obviously, this is a big problem, but we thought we'd start by breaking down the complex protein structures into three main classes: unstructured regions, alpha helices, and beta strands. To do this, we turned to the Protein Data Bank, the standard online repository for protein structures. We downloaded 6,000 protein sequences corresponding to 38,000 unstructured regions, 61,000 alpha helices, and 33,000 beta strands. We first engaged in exploratory data analyses of these sequences to see if we could detect any trends in amino acid usage among these structural subtypes. Turns out that some amino acids like leucine and glycine show different usage patterns, so we thought we might be able to use this as a good predictor. However, a simple principal component analysis wasn't enough to accurately classify all these different kinds of structures. Our next idea, a random forest model, turned out to be better for classifying helices, but wasn't so good at picking out the unstructured sequences. So instead of telling our models to rely on the amino acid composition, we turned to more unbiased methods that took into account the actual sequences of amino acids. Naive Bayes and PCA analyses each turned out to be good for two of the three substructure types. In the end, though, we finally settled on a support vector machine as the best predictor of all three structural types. So what did we learn? Even with these simple methods, we were able to predict the structures of many primary sequences. We imagined that these methods could be greatly improved upon, though, but would make for a good starting point for future protein prediction problems.